Good morning. Today I am going to talk on genome sequencing and whole genome sequencing tuberculosis, culture and drug susceptibility, that is DST. Genome sequencing is a powerful tool in the study of Mycobacterium tuberculosis, that is a M tuberculosis, providing critical insight into its genetic makeup, drug resistant mechanism and transmission. So, transmission patterns, whole genome sequencing, which is known as a WGS, is an advanced technique that enables us comprehensive analysis of the entire genome of Mycobacterium tuberculosis, playing a crucial role in tuberculosis, that is TB, diagnostic, treatment, strategy and epidemiological studies. So let's introduce to genome sequencing. Genome sequencing refers to determine the complete DNA sequence of an organism's genome at a single time. It helps in identifying genetic variation, mutation and evolutionary patterns in microbial pathogens, including Mycobacterium tuberculosis. Types of genome sequencing. First, targeted sequencing. It focuses on the specific gene of interest, such as resistant associate with genes. Second, it's a whole genome sequencing, which is also known as WGS. Analyze the entire genetic material of an organism, providing a complete picture of mutation and genetic variation. Whole genome sequencing, WGS, and it's important in tuberculosis. Whole genome sequences is the most comprehensive genomic approach for studying Mycobacterium tuberculosis and is revolutionizing TB diagnostic and research. It also provides detailed insight into drug resistant mechanism by which identifying mutation in genes associated with drug susceptibility, transmission dynamics by tracking genetic variation in TB strain, evolutionary pattern of Mycobacterium tuberculosis, which help in vaccine and drug development. Advantage of whole genome sequencing over conventional drug susceptibility test and LPA, that is a line probe assay. Role of whole genome sequencing in TB culture and DST, drug susceptibility test. Whole genome sequencing is integrated into TB culture and drug susceptibility tested in the following ways. 1. identifying the drug resistant mutation, rifampicin resistant mutation in the RPOB gene, in isoniazid resistant mutation in CATG and INHA genes, in fluoroquinolone resistant mutation in GYRA-A, GYRA and GYRA-B, that is GYRB genes. In second line, injectable drug resistant mutation in RRS and EIS gene, bedaquiline and linezolid resistant mutation in MMP, R5, ATPE, RP, IC, RRL genes. It also causes rapid turnover time for DST. Traditional DST, that is drug susceptibility test methods, takes weeks to months because of Mycobacterium tuberculosis grow slowly. Whole genome sequences can detect resistant mutation within 3 to 7 days directly from the culture or even clinical specimen. Application in TB control program. Epidemiological surveillance. It helps in tracking out TB outbreak and identifying transmission chain. It also tailors the treatment regime, allow for a personalized TB treatment based on resistant profile, monitoring emerging drug resistance. It helps to detect new mutation associated with resistance before they become widespread. And fourth, whole genome sequencing workflow in TB culture and DST. Step 1, sample collection, sputum, bronchoalveolar lavage, tissue samples are collected and cultured on LJ media, that is Lawrence-Jensen media, or MIGET, that is a Mycobacterium growth indicator tube. Step 2, DNA extraction. DNA is extracted from microtuberculosis isolate using specialized method such as bead beating or enzymatic lysis. Step 3, library preparation. The extracted DNA is fragmented and adapters are added to prepare it for sequencing. Step 4 is a sequencing next generation sequence, that is NGS. Platforms such as Illumina, Oxford, Nanopore or PassBio are used to generate sequencing data. And in step 5, bioinformatics analysis. The raw sequencing data is aligned against the H37RV reference genome of Mycobacterium tuberculosis. Mutation associated with drug resistance are identified using database like TB Profiler, Pyrus SC, and Microbe. And in step 6, interpretation and reporting. Resistant profiles are used to guide clinical decision and inform public health intervention. Challenges and limitation of whole genome sequencing in TB drug susceptibility test. Future direction and conclusion. WGS, that is the whole genome sequencing, is transforming TB diagnostic and surveillance, providing rapid and comprehensive insight into drug resistance and transmission dynamic. The future advancement include point of care whole genome sequencing platform for real-time resistance detection. It can be integrated with artificial intelligence and machine learning for improved mutation prediction. Expansion of whole genome sequencing in high burden setting to improve treatment outcome. And in the final thought, whole genome sequencing is the future of TB diagnostic, offering a faster, more accurate alternative to conventional drug susceptibility tests, that DST. While challenges remain, the increasing affordability and accessibility of sequencing technologies are making WGS, the whole genome sequencing, a crucial tool in the fight against tuberculosis.